Hello friends, welcome to my channel Pharmagyan. Friends, in this channel you can learn about pharmaceutical guidelines, pharmaceutical industrial practices, processes, documentation practices, and many more about pharmaceutical industries. Friends, please don't forget to like, subscribe and share my channel Pharmagyan, and press bell icon given in bottom right corner to get latest updates directly in your notification panel when I upload new videos. So next we will discuss about the video.
Part 7 of 8. Determination of the amount of residue. Introduction. This section provides a practical guidance for the determination of the amount of residue in cleaned equipment based on the requirements from regulatory authorities for and current guidelines on analytical to examples of sampling methods and the appropriate use of analytical methods. Determination of the amount of residue. Introduction. This section provides a practical guidance for the determination of the amount of residue in cleaned equipment based on the requirements from regulatory authorities for and current guidelines on analytical validation. 5 Specific requirements for the validation of analytical and sampling methods for cleaning validation purposes are provided in this section, in addition to examples of sampling methods and the appropriate use of analytical methods. The carryover acceptance limit, MPA, is a calculated figure that represents the specification limit for the equipment cleanliness. See section 4.0, acceptance limits. However, the determination of the actual amount of residue, M, remaining in the equipment following cleaning must be achieved using appropriate methods, that is for both the sampling method and the quantitation of the contaminant in the sample. Since the decision on the acceptable cleanliness of the equipment bears a potential risk to product quality, the methods used for the determination of M must be validated. 1 And the specificity, sensitivity and recovery of the methods should be determined as a minimum. Determination of the amount of residue, continued. Validation requirements. General. The requirements for analytical method validation are defined in ICHQ 2R1, Validation of Analytical Procedures, Text and Methodology, November 2005. There are 4 types of analytical methods with principally different validation requirements. These are identification tests, tests for impurities, both quantitative and limit tests, and assay tests. The validation requirements for each method type are shown Table 1. The list should be considered typical for the aforementioned analytical procedures. However, exceptions should be dealt with on a case-by-case basis. It should be noted that robustness is not listed in the table and should be considered at an appropriate stage in the development of the analytical procedure. In practice, it is usually possible to design the experimental work such that the appropriate validation characteristics can be considered simultaneously to provide a sound, overall knowledge of the capabilities of the analytical procedure, for instance, specificity, linearity, range, accuracy and precision. B. Validation requirements, continued. The validation of an analytical method should occur in compliance with pre-established acceptance criteria that should be documented in a written general policy or validation plan. However, there should be one validation report per validated method that summarizes the specific results. Characteristic. Type of analytical procedure. Identification. Testing for impurities. Assay. Quantitative. Validation requirements, continued. The requirements for testing for impurities are typically employed for the validation of analytical methods specific to cleaning validation. The requirements for quantitative testing for impurities can apply, for example, in cases where a method should be suitable for several possible acceptance limits and therefore quantitation of the residue over a certain range may be necessary. For example, the measured amount of residue M must be compared with acceptance limits between 5 and 750 grams/equipment. This is possible when the method will be used for several changeovers. The requirements for limit testing for impurities can apply, for example, in cases where the method should be suitable for one specific acceptance limit. For example, the measured M must be compared with M per 105 grams/equipment. B. Validation requirements, continued. Analytical method validation for cleaning validation. In the following sections, aspects of analytical method validation specific to cleaning validation are emphasized. For further details, refer to ICHQ2, R1. Specificity is a basic requirement for all analytical methods. See Table 1. However, in the case of cleaning validation, it may occur that not all potential impurities are clearly specified. It is important to note that in such a situation, a specific method may not always detect all impurities. Studies should be performed to characterize the unknown impurities, develop and validate suitable analytical methods. However, this can be an unacceptably time-consuming task. In this case, a method that detects all potential impurities together can be suitable, even when it is not specific for each of the impurities. For example, in a situation where only non-volatile impurities occur, a dry residue determination method that is specific for the sum of non-volatile impurities could be used, provided that the validation requirements according to Table 1 are satisfied. In order to consider the equipment acceptable for use, it must be assumed that the dry residue consists of the worst-case impurity, most toxic, most active, etc. In some cases, a combination of several methods can achieve the necessary specificity. Analytical method validation for cleaning validation, continued. After the completion of a cleaning validation study, an unspecific method, for example dry residue, may be used for the routine verification of equipment cleaned by the validated cleaning procedure, provided that it is shown that the unspecific method is suitable for the intended purpose. If possible, the sensitivity of impurity detection for cleaning validation should be determined for both the sampling and analytical methods together. See Section 7.2.4. B. Validation requirements, continued. Detection and quantitation limits. Measured values below limit of quantification, LIQ, should be reported as the LIQ value, worst case approach. For example, if the LIQ is 10 mg/L, the measured blank is 7 mg/L and the measured residue amount is 3 mg/L, the reported value for the sample should be equal to the LIQ, that is 10 mg/L. Usually it can be assumed that, for quantitative impurity determination, the LIQ should approximately be 0.5 of the specification, that is for cleaning validation 0.5 of the acceptance limit or lower. LIQ should never be higher than the acceptance limit. In the following sections, 3 methods of LIQ determination are outlined. Based on visual evaluation. Visual evaluation may be used for non-instrumental methods but may also be used with instrumental methods. Frequently this approach is used for TLC. Detection and quantitation limits, continued. Based on signal-to-noise approach. This approach can only be applied to analytical procedures which exhibit baseline noise, for example GC, HPLC. A signal-to-noise ratio, S/N, between 3 or 2 to 1 is generally considered acceptable for estimating the detection limit, LID, and a typical ratio for acceptable quantitation limit is 10 to 1, LOC. The value for S/N can be calculated according to Equation 1 and Figure 1. Equation 1 equals 2xH where h is the height of the peak from the mean baseline. Hn is the maximum deviation of the baseline within the range of 5 to 20-fold width of peak at half height. Detection and quantitation limits, continued. Figure 1. Detection limit based on signal-to-noise approach. Detection and quantitation limits, continued. Based on the standard deviation of the response and the slope, the detection limit may be expressed by Equation 2 and the quantitation limit by Equation 3. Equation 2 equals 3.3x. Equation 3 equals 10x. Determination of recovery. If possible, the recovery of impurity detection for cleaning validation should be determined for the sampling and analytical methods together, at least for recovery and sensitivity. Limit of quantitation, LQ, or limit of detection, LD. This can be achieved, for example, by spiking a surface equivalent to the equipment surface, for example material, Polish grad.d.t, with different known amounts of the impurity. The impurity can then be recovered and analyzed using the same sampling and analytical methods that will be used for the cleaning validation study. The overall results from this procedure are then compared to criteria for detection or quantitation limits as defined in ICHQ 2R1. Validation of the limits may be achieved by the analysis of samples known to be near at the limits. The measured results are then compared to the actual amount applied to the surface. The recovery is typically determined during the accuracy determination and should be reported as a percentage of the known applied amount of the impurity. Determination of recovery. As an example, quantitative impurity determination recoveries of 90% are usually regarded acceptable. For cleaning validation, recoveries of 90% do not need to be taken into account for the calculation of the true value for M. Recoveries of 90% must be included in the calculation for M, see equation 4, and recoveries of 50% should be omitted. Equation 4e. Where M, true value for the amount of residue remaining in the equipment after cleaning. Mres, the measured amount of residue, sampling and then analytical measurement, are recovery in percent divided by 100. For example, for 75%, 75 100ths equals 0.75. Validation requirements for quantitative testing of impurities. The requirements for the validation of quantitative testing of impurities according to ICHQ 2R1 are shown in Table 2, including proposed acceptance criteria, as an example only. Alternative acceptance criteria may be established based on sound scientific rationale. It is important to note that the summarized requirements should be used for the validation of quantitative testing for impurities during cleaning validation studies. Validation of quantitative testing for impurities is usually applied when the analytical method will be used for several specifications of the residue amount in the equipment. The lowest foreseen acceptance limit is referred to as MPer-min and the highest limit as MPer-max in Table 2. For only one specific acceptance limit, normally limit testing for impurities and the corresponding validation of the analytical method is sufficient. If the validation of quantitative testing for impurities will be used for one specific acceptance limit, then MPer-min equals MPer-max equals MPer. For the experimental work described in Table 2, the samples can be spiked with appropriate levels of the impurities, when standards are available, or compared with another well-characterized procedure, when standards are not available, to obtain the true value of the analyte concentration.